So thank you, Sarah, for the introduction, and also thank you and Ronnie for the invitation to summarize the 2012 BGRI Technical Workshop meeting. And also I'd like to take this opportunity to thank Dr. Wangquan Chen, who's organized this meeting and invited us and welcomed us to Beijing. And it's quite an interesting gathering of rust pathologists, breeders, geneticists, funders, farmers, even people from the private sector. It's quite a neat group. And over the last 4 days, we've shared our research, we've enhanced our networks and collaborations, and we've even walked the Great Wall together. And so for this summary, what I'd like to do is to divide it into two components. And first, just talk about what we have learned, and then conclude by addressing where we are going. And so to start off in what we have learned, in the keynote address, Michael Baum was discussing how even today, 1 billion people go hungry every night. And so we still have significant agricultural issues right now, and it's only going to get worse, as many of the speakers were indicating, in the gap between yield production and need for those resources. And so here's a slide from Michael's presentation showing the stripe rust epidemics worldwide and how devastating these can be. And one thing that he really emphasized in his presentation was that stripe rust was an international pathogen, thus needing this international collaboration, all of us coming together to try and solve a problem. So that was how he started off this address. And we continued learning more about the pathogen. Really inspiring was Dave Hodson's work in showing how much progress has been made in the construction of a wheat rust toolbox. Also with Les Sabo's work, demonstrated in many of the posters, and developing a rapid molecular diagnostic assay for the pathogen. And so we have several new tools that are enhancing our pathotyping capacity. Also Morgan's Hofmahler in establishing the Global Rust Reference Center. And so global, overall, worldwide, we're seeing really a renaissance in the ability to pathotype, both at the phenotypic level and the genotypic level, rust pathogens. Unfortunately, we did have a panel come in and talk about how maybe the pathogen is more dangerous than we have been thinking. And so the Barbary panel was constructed of members shown from across the world, and they highlighted that in the Pacific Northwest, in Russia, in Sweden, even in Central Asia, Puccinia graminis is going through the sexual cycle. And then also Dr. Kong presented I think the first results showing actual Puccinia strophomonas infection in the field in China. And so these sexual cycles may be relevant for agriculture. But I think that the question that's pointed out in their slide, point number 2, what role, if any, do these species play in rust epidemiology today, is still a very relevant question that needs to be addressed. We know this is going on. How much of a role is this playing in agriculture? And if so, what should we do about it? Also, John Rathjen was describing some more information about the pathogen and pathogen diversity, how diverse effectors are. And so even among isolates, we see this great variability that could be quite dangerous. And so the situation could be described as quite dire. We have diverse pathogens spread across the world, whether it be in Kenya, in stem rust epidemics in 2007, or in Ethiopia, stripe rust epidemics in 2010. It doesn't really look good. And as Norman Borlaug would say, we need to get to work. But the truth is, there has been a very substantial amount of work that's been taking place. And so some really exciting results were presented showing the distribution of resistant varieties to different countries that are right in line to possibly receive UG99 next, and also have been suffering from stripe rust. And so I'd like to highlight the Nepal group also has, even within their national program, released 14 resistant varieties. And this work was highlighted by their receipt of the Gene Stewardship Award. And I think this is very important to set a precedent for other national programs to really step up to the challenge of developing resistant varieties and getting them to the farmers. And so their variety VJ is on 5% of the wheat acreage in Nepal. And this is just an unbelievable achievement when you think that just in 2005, Norman Borlaug was sounding the alarm about UG99 stem rust. Just 7 years later, resistant varieties are already in farmers' fields at a low percentage, but still, this is quite remarkable. And so much of this has been due to the USAID funded work through CIMMYT and ICARDA and in getting those resistant varieties out. And also, Indu Sharma was commenting about how in India, wheat variety replacement is essential. And the two farmers, Mr. Singh Man and also Mrs. Makita, were emphasizing the importance of wheat variety replacement and how successful that has been for them on their farms in order to mitigate against rust. And so into the future, we really need to focus on wheat variety replacement as well. And so when you look at these varieties, you can say, well, where did they come from? We just pull them out of a hat? Or, in this quite remarkable achievement, the dogma that has been, been shown by many of the talks is, Ravi Singh's program in, using shuttle breeding and the bulk phenotypic selection of wheat lines that are resistant to rust, also high yielding, also resistant to abiotic stresses, and this results in the development of these new cultivars. And when you look at the progress that they've made, it is, I think, unprecedented. And so you can see in this graph here of Ravi's, back in 2005, you have the distribution of breeding lines, and across the x-axis is grain yield. And you can see the improvement in 2010, 11, and 12. And I'd like to make the point again that the x-axis is not rust resistance, it's grain yield. And so in those 2011 and 2012 varieties, not only are they almost all rust resistant with complex genetic inheritance, they are very high yielding grain. And so I think a more accurate picture of what's really going on in this process is you have these excellent scientists with a really good breeding eye, and they can see in the field which plants are high yielding and picking those. And so without really the expertise of Ravi and Julio Huerta, I don't think this amount of progress could really have been achieved. And so I think that the global wheat community and farmers worldwide really are in debt to them. And so throughout the conference, in addition to having these cultivars with adult plant resistance, we've begun to understand the characterization of this resistance. And so here's just a slide from Godwin Macharia showing QTL, or associated markers for stem rust resistance. And as many of us have perused the posters, you can see so many new genes, so many new QTLs to all the various rusts. And so, even Mark Sorrells' group has constructed a UG99 and rust resistance consensus map. And so there's really a plethora of new information coming on that are providing us with the tools that the breeders need. And so when Mike Pumphrey was discussing this, he showed the slide that only a few years ago, the sources of resistance were few and far between, and many of them weren't really accessible. But now the situation has changed dramatically. We have a very large number of genes. The breeders have plenty of genes to choose from. We're even trying to stack genes on single chromosome arms and make progress in this regard. And so it's really a game-changing event that's taken place. This DRRW project and also contributions by national programs have really answered the threat of UG99. Many new technologies were discussed, and so when Catherine was here discussing the progress on the wheat genome sequence, and though it's 5 times as large as the human genome sequence, even at the current state, there's still plenty of information that can be pulled from those genomic tools. And so her example of the rapid cloning of the gene SV2 can be easily applied to rust resistance genes. And so this is an exciting development. Also, the graduate student, Wei Lu from Northwest A&F University, presented her work on the rapid cloning of the gene YR10. And so we're seeing a renaissance of the ability to go in and clone these genes and figure out what they're doing. And particularly inspirational was the presentation by Brand Wolf and his colleagues at the Sainsbury Laboratory. And so in their development of a pipeline for the rapid cloning of multiple resistance genes. And so this work could be key in the development of a transgenic cassette of multiple resistance genes that could be used in breeding to provide breeders with a single locus to provide durable resistance. And so hopefully that work will come to fruition. Also, Beat Keller demonstrated the successful transformation of LR34 into barley. And this is particularly going to be useful for the understanding of the functional, how LR34 is functioning in barley. And so having it in that diploid background could be useful. And so understanding what is making LR34 durable could be useful for the future in order to find more durable resistance. There's a few talks that discussed effectoromics, and so here's just a slide from Diane Saunders and showing how she's done a comparative analysis of effectors in order to identify conserved effectors that could be targeted for durable resistance genes to interact with. And so there's a number of different studies coming together on this in this area. And so even in the last year, we've seen the first true avirulence effectors from the Puccina graminis wheat stem rust pathogen. And so it seems like there's a critical mass of research and researchers on this topic, and hopefully we're going to see some new developments in the future with regards to using effectors to find resistance genes in diverse germplasm, using effectors to even design new resistance genes. And so it's really exciting to be involved in research at this time. Another one of our graduate student presentations highlighted a new technique to sort Hostoria. And so this could help allow for the characterization of effectors even faster. And really inspirational was the work of Dr. Lau, and showing that a transformation of wheat with an antifungal protein and an antibody that interacts with a fungus can provide resistance to a disease that's much more complex and difficult than the rust pathogens. And so I think this can inspire us to think outside of the box, think about new ways to find resistance and to stop stem rust and the other rusts. Of course, many of these new technologies are going to rely on genetic transformation, and so in the biotech panel we heard about the pitfalls and also the promises of GM wheat. And so, and really, what I really took home from that was in Peggy's presentation, she really emphasized the importance of public and private partnerships. And so the private companies are coming up with these new technologies. Those technologies need to be utilized by the researchers, not just to make genes to give for use in agriculture, but for the functional validation of genes in research. And so those exciting developments are there as well. And so another example of a good public-private partnership is the Monsanto Bichel Borlaug Scholars Program. And so when Ed Runge was here explaining this, and we learned of all the exciting developments, and Godwin Macharia was a recipient who presented here, and this is really leading the way with hopefully a sustainable form of training and international development. And then Amor was discussing all of the training capabilities and activities going on at CIMMYT. And not only at CIMMYT, but I think around across the world, many graduate students are being trained. Many scientists are visiting pathology labs and genetics labs to receive the training that they need. And so this training is really, as I said before, providing a renaissance of rust pathology and breeding for resistance. And though we have all this training and all these new scientists coming, I think it's unfortunate that we've seen the retirement of a very key player in rust pathology and breeding for resistance of Professor Colin Wellings. And I think we hope that in the future he'll still provide his expertise internationally, much as Dr. Bob McIntosh has, And in the future, we hope to see more of him. But he's really leaving a big gap, especially in his work with stripe rust in Asia and around the world. Also, we've seen the premature death of Sam Colonzo. And this was a man who was a good friend to many of us. And his work in the International Screening Nursery in Kenya will be missed and also needed. And so these are big gaps that are missing. But hopefully the training opportunities and what has been going on internationally will provide the new scientists to come in and help fill those gaps. So that's kind of an overall summary of what we have learned. So I think you can divide that into three different key components. And so one, the pathology. And so we've seen these new tools being developed, the molecular diagnostics, the wheat rust toolbox. At the same time, we're starting to understand more about how pathogen diversity can be more of a problem. And so it's kind of, we're getting mixed feelings there, but we are making progress in understanding what's going on. A second point of what we have learned is with regards to the host and breeding for resistance. And so we've seen the first generation of UG99 resistant varieties make it out into farmers' fields. We've also seen an unprecedented amount of resistance genes and genetic stocks being produced that breeders can use. And thirdly, we've seen just a renaissance in training, and also we really emphasize that when Amor was talking about training, he had pointed out that in recent years, CIM has been training more and more women. And so also a really good indication of enhancing the role of women in agriculture as a whole would be when Haven Lay was giving her talk. She was saying that around 70% of farmers around the world are women. And so if we really want to make a difference efficiently, we need to be having training of women, because they're really the ones who are involved. And so at this meeting, there are 238 attendees, and 66 of those are women. And so this is the highest amount of women that we've seen at any BGRI conference. And so hopefully we're setting a precedence for the future in trying to fill the gap in women in agricultural research. So with regards to where we are going, I'd like to start this by giving a brief historical example. And I think we can learn from this historical example for the future. And so if you'll bear with me, I'm going to put up a slide that might be familiar to many of you. So on the x-axis is time, starting in about 1918 through 2008. And on the y-axis is U.S. percent wheat loss to stem rust. So across the whole U.S., production loss to stem rust. And so in epidemic years, you saw 10 to 20%, but that was across the whole United States. And in certain regions that are the size of many countries, yield losses were as high as 50%. And what was done in the past in order to stop stem rust was initially barberry eradication. And so we know that this eradication program went through until about 1980. However, much of the barberries around wheat fields were eradicated as early as 1930, but there were still stem rust epidemics. And those epidemics were associated with this boom and bust cycle. And so resistant varieties like Ceres and Triumph were deployed, new virulent races like race 56 arose and caused a bust in the resistance. Thatcher was deployed, and then race 56, race 15B came in and caused epidemics. And in the 1950s and 60s, there was this big movement to try and stop stem rust. And so there was a whole bunch of international work, funding that was being provided in order to develop resistance. And so some of the derivatives of that resistance were Chris, which is a Minnesota cultivar from that time period. But also in Kenya, varieties were developed. We call them the old Kenya varieties. And those varieties, and also Chris, are still resistant to this day to stem rust, even UG99. And so even at that time period, you can see that there were investments in East Africa. This is a building built in collaboration with Canadian scientists in Njoro that was used to study rust. Now what happened was, though breeding for resistance continued after that time, it didn't stop, but it was relaxed. And so the emphasis on this complex, durable resistance kind of got forgotten over time. Resistance became to be relied more upon single major genes worldwide, like SR31. And we know that all of a sudden we have this UG99 threat. And so I'd really call us, as we look forward and where we're going from here, is to learn from that example. We've seen these epidemics, this severe threat of UG99 and stem rest. The resistance varieties are here, they're being increased, but let's not just leave it at that. We need to increase the sustainability of breeding for durable resistance for the long haul. And so this is a building built recently by the DRRW project in Njoro, Kenya. And the question is this going to be kind of like a relic in 30 years, or is this going to be something that's maintained and used for the long term? And here's the objective statement of the BGRI. And I think it's appropriate at this point to read it. And it says, the BGRI has the overarching objective of systematically reducing the world's vulnerability to stem, yellow, and leaf rust of wheat, and facilitating the evolution of a sustainable international system to contain the threat of wheat rust and continue the enhancements in productivity required to withstand future global threats to wheat. And so the key point here is we can't just stop and move on to other things. This research needs to continue. We need to make sure that this great relationship that we have between pathologists, breeders, funders, farmers, continues into the future. If we start to break those bonds, break the bond between pathology and breeding, the breeding is going to go on and kind of forget about this whole durable resistance thing. And so it's absolutely key that 30, or maybe even less than that, 30 years from now, we don't end up in the same situation that we were just a few years ago. The final slide I'd like to show is this. And so, the really exciting results of the new resistant varieties being deployed. The immediate concern is wheat variety replacement. And as the farmers who were here were saying, and I think it was Mrs. Makita who said the resistant varieties need to continue. And I think she hit the point exactly. We have resistant varieties, but they need to continue for the long term. And with that, I'd like to thank you.